Okay, so worksheet number 4. Now remember, with these worksheets, it is best for you to go ahead and attempt to do the worksheet and then to, of course, listen to the feedback from that. Okay, that is going to be the best means of you learning the material, okay, and taking the biggest benefit away from these worksheets. So I would encourage you, if you haven't worked through this worksheet, to go ahead and stop right now and to do the worksheet and then to come back and listen to this recording. Okay, so go do that. Right now, having done that, when we think about this worksheet, we're looking at actually material that is in really lecture 6. And in lecture 6, we're talking about not only carbohydrate metabolism, but the regulation of carbohydrate metabolism. We have now covered pathways like glycolysis and gluconeogenesis and glycogen synthesis and glycogen breakdown, right? And those are very important pathways. And it's also very important that they be regulated. Right? And the regulation of these pathways is done differently in different tissues, as I have tried to illustrate to you in lecture 6. Okay, so we've talked about, of course, the liver, we've talked about skeletal muscle, we've talked about cardiac tissues, right? And we see that in each of those tissues there are small nuances in terms of how these pathways are regulated that are different from one another, right? Now, that's really actually quite fascinating. Okay, from a biochemistry standpoint, it is really quite fascinating when we look at this. However, it is also very complex. And it is sometimes difficult then to try and maintain order in our brain as we go through this material. In particular, for a student, the first time you've gone through it, it can be very difficult to organize the differences in terms of regulation of glycolysis and gluconeogenesis and glycogen breakdown and glycogen synthesis, and then add on top of that the tissue specific regulation. Okay, so what I want to do in this worksheet is give you a potential means to organize in your mind the regulation of glycogen, I'm not sorry, glycogen, but glycolysis and gluconeogenesis in the liver cell. Okay, so we're not going to go over all of it. Okay, we're going to talk about the liver cell, and we're going to think about glycolysis and gluconeogenesis and how they are reciprocally regulated within this tissue. Okay, now gluconeogenesis, just so we know, isn't really found in the cardiac tissue and isn't really found in skeletal muscle, right? So we don't really talk about the regulation of gluconeogenesis outside of the liver. So this is a very liver, quote unquote, liver specific discussion. Okay, and of course glycolysis is found in skeletal muscle and is found in cardiac muscle, so we can talk about the regulation of that, but not in opposition to or in reciprocal to gluconeogenesis. Okay, so just remember that, right? That gluconeogenesis is a pathway that's found pretty much in the liver. Okay. So anyway, what you're looking at here is this flow chart, and sometimes flow charts are really good ways for students and faculty members, okay, professors, to organize their thinking and organize their thoughts, in particular on subjects that are complex and have multiple steps, okay? And so that's what we're looking at here. We're looking at two different conditions. We're thinking here about blood glucose and how it could drop, okay, and then what that will result in terms of a change in metabolism within the liver. And then, of course, on the other side of this would be a rise in blood glucose, okay? And we'll look at these separately. And then we have these terms up here, okay? I listed them all here. They're, of course, listed on the worksheet. And we're going to put those terms in these boxes, and we want to modify them, right? So, for instance, will it rise? Will it go down? Will it be inhibited? Will it be activated? Will it be phosphorylated? Will it be dephosphorylated? Something along those lines, these terms, okay? And then that's in what we call activity 1. It says, in fact, it says, in this activity, you will need to fill in empty boxes with the following terms based on the sequence of events that occurs. If you like, you can also modify each term with modifiers like, and then there's this list of them, okay? And then here are our terms, okay? So when we think about this, let's go ahead and start on the left, all right? And we have a drop in blood glucose, okay? So we're in what would be considered a fasted state. Blood glucose is steadily declining in that case. And in a mammalian system, that is going to lead to the production of and excretion of the hormone glucagon, okay? So we're going to go like this. We will put here, this will be our very first thing, glucagon will increase, okay? And so I'll just put an up arrow and I'm going to cross that out. Glucagon increases. Right, and that happens in the cells of the pancreas. Okay, now understand something, that glucagon is a hormone that doesn't have a lot of targets within the human body. Okay, and in a mammalian system, there are glucagon receptors on liver cells. Okay, but there are not glucagon receptors on every cell in our body. Okay, so when we think about glucagon, it's going to primarily affect the physiology and the metabolism of the liver. Okay, so just remember that. Don't find glucagon receptors on skeletal muscle. Okay, so glucagon increases. Now it's going to then bind to, and these are not terms there, but I'm going to go ahead and fill in some of these gaps, okay, for us, just as just to help you out in your studies and just to go over this stuff again. But glucagon is going to bind to the glucagon receptor. The glucagon receptor is what we call a G-coupled G protein coupled receptor. Okay, and that G protein is going to, when glucagon binds to it, it's going to dissociate, okay, and one of its subunits is going to diffuse laterally in the membrane, and it's going to bind to and activate the enzyme adenylate cyclase. And adenylate cyclase, if you remember from our discussion in lecture 6, is going to make cyclic AMP. Okay, cyclic AMP is a small molecule messenger, and it is going to then start to accumulate in the liver cell. And when it accumulates in the liver cell, it will bind to and activate pKa. Now, the details of that are, it's going to bind to the regulatory subunit of pKa, it's going to cause a conformational change in that, and then that catalytic subunit is going to be released. Okay, so what we're going to have here, and we do have pKa up here, we have here then pKa is going to be activated. And there's our modifying term. Okay, so glucagon rises, pKa is activated. Okay, now when pKa is activated, pKa stands for protein kinase A. It can also go by CAPK, cyclic AMP dependent protein kinase. Okay, and then you get to this whole really weird, sentence, pKa, aka CAPK. Okay, right? So, I mean, that was, I like to have fun with those kind of things. I know I'm a geek, but anyway, all right, so the pKa is activated. It is a kinase. It is going to phosphorylate things, okay? And so what we're going to do here is we're going to just say that PFK2 and FBPase2 are going to be phosphorylated, okay? So I'm going to put here PFK2 backslash FBPase2 are going to be phosphorylated by the activated pKa. Now, I'm going to go ahead and, I mean, the modifying term will be phosphorylated, but I'm just going to go ahead and put a phosphate group, and I'm going to put it coming off of PFK2. Now, remember, this is a protein that we say is bifunctional, okay? It has two different domains. There is a kinase domain, which is the PFK2 domain, and then there's the phosphatase domain, which is the FBPase2 domain, okay? And the PFK2 domain, or the kinase domain, makes fructose 2,6-bisphosphate, okay? And the FBPase2 domain, or the phosphatase domain, makes, or not makes, but breaks down F2,6BP, okay? And so I'm just going to draw a little something here. When this gets phosphorylated in the liver cell, okay, it gets phosphorylated on the kinase domain, and that activates FBPase2, the phosphatase domain. Okay, and what's going to be the result of that is that then F2,6BP will decrease. Okay, so it will go down. Right? And you say, yes, that's right. Fructose 2,6-bisphosphate goes down. What is that again? Okay, so we got to keep this straight, because there is F16BP, fructose 1,6-bisphosphate. Now, fructose 1,6-bisphosphate is not fructose 2,6-bisphosphate. They are very, very similar to each other. The only difference between the two is that there's a phosphate on the second carbon of F2,6BP rather than on the first carbon, which is found in F16BP. Okay, now F2,6BP is an activator of PFK1, and an inhibitor of FBPase1. And you say, ah, yes. What are those again? PFK1, okay, remember, is a glycolytic enzyme. Okay, it is one of the key regulatory points in glycolysis, and it can be activated when fructose 2,6-bisphosphate levels are rising. FBPase1, on the other hand, is an enzyme found in gluconeogenesis, is one of the key regulatory steps in that, and it will be inhibited as F26BP rise. Okay, so we see here that we are seeing a falling of F26BP, which means this, that we can say FBPase1 is going to be, and I'm just going to put a little plus sign here, it's going to be activated. Okay, all right, so we can actually come up here and cross this off. Now, you might look at this and say, man, it sounds so similar. Why do we call it FBPase1 and FBPase2? And the answer is because. Okay, so I know they're really similar, and I hate that they're so similar. Okay, they do very similar things though, and that's why the names are so similar. FBPase1 is going to convert fructose 1,6-bisphosphate to fructose 6-phosphate. Okay, FBPase2 is going to convert fructose 2,6-bisphosphate to fructose 6-phosphate. So if we were to look up here, what this would be doing is it would be taking F26BP, oops, I meant to draw a 6 there, 6BP, and it's going to convert it to fructose 6-phosphate. Okay, and again, this is a regulatory molecule. It's not found in glycolysis. It's not found in gluconeogenesis. It's just a regulatory molecule. Okay. This, on the other hand, come down here. This is going to take fructose 1,6-bisphosphate, which is found in gluconeogenesis and glycolytic pathway, and it's going to convert it to fructose 6-phosphate. Okay. And then in gluconeogenesis, this is going to be converted to glucose 6-phosphate. And then we need another enzyme to convert that to glucose, right? Remember that, okay? Right, so all of this to say that when we get back here, when we got an activated FBPase1, just as I'm showing here in green, that means that gluconeogenesis flux is going to increase, okay? And that is all the result of a drop in blood glucose. Okay, now that hopefully makes sense, because what you see then here is gluconeogenic flux increasing. That's going to make glucose. And the liver, one of the jobs of the liver is, of course, to make glucose and to bring glucose levels up, okay, in the bloodstream. Okay, and that needs to happen if blood glucose drops. Okay. All right, so let's go over to our right now, and let's go ahead and do blood glucose rises. So this would be the opposite. All right, this is a person in the fed state, and the blood glucose starts to rise as their body absorbs the nutrients that they are, of course, eating. Okay, in particular, carbohydrate-rich meal. Okay, now in that case, the very first thing that happens is that insulin, not glucagon, but insulin is going to increase. Okay, so we'll put insulin is on the rise in this case. Okay, and insulin is a hormone that has really widespread effects in the body. It doesn't just affect liver metabolism, though that's the only thing we're going to look at here in this activity. But insulin has receptors found on many different tissue types, including skeletal muscle, including cardiac tissue, okay, including adipose tissue. All right, so there's lots and lots of different tissues that have insulin receptors on them. Okay, now again, we're not going to fill in all the details here. I will go ahead and talk about the details, but not all the details are shown in these boxes. But insulin, when it binds to its receptor, that's a tyrosine kinase receptor, it undergoes an autophosphorylation event, and that autophosphorylation allows for the recruitment of IRS, insulin receptor substrate, okay, to the membrane. And when it is recruited to the membrane, it will also recruit another enzyme called PI3K, or phosphoinositol 3 kinase. Okay, PI3 kinase would be another name that it goes by. And again, not one of our terms, but I'm going to go ahead and fill this in. Now, if you remember, going back to insulin signaling, PI3K is going to convert PIP2, phosphoinositol, phosphoinositol 4,3, I think it's 4,5 bisphosphate. It's going to convert PIP2 to PIP3. This is PI3 kinase, and PIP3 is phosphoinositol 3,4,5 phosphate. Okay, these are lipids in the membrane. Okay, it goes from PIP2 to PIP3. It's just essentially phosphorylating PIP2 to make it PIP3. It's just adding one more phosphate to it. Okay, and like I said, they're lipids. They're phospholipids actually in the membrane, and PI3P will then start to accumulate in the inner leaflet of these cells that have insulin signaling on them. PI3P or PIP3, I'm sorry, will then recruit, okay, PDK and AKT to the membrane, meaning they will start to bind to the membrane, okay? And it's really AKT that we care about in our little discussion here. And what's going to happen here is this, that AKT is going to be activated. Okay, so I'm going to go ahead and put a little plus sign here to mean that it's activated. Okay, now AKT is kind of the equivalent of pKa. Okay, pKa goes out and has vast effects. AKT will go out and have vast effects. Okay, and AKT is actually more complicated in terms of what it can and does do in the cell. But one of the things that we know that it will do is that it will activate phosphoprotein phosphatase. Okay, so I'm going to go ahead and put here the activation, the downstream activation of phosphoprotein phosphatase. Now, if you're feeling like there's some gaps here, okay, in our little presentation of this, you probably are right. There are some gaps here. We're not leaving some things out in terms of how does AKT activate this, but let's just, for now, ignore those gaps and just say that this is going to be activated. Okay. All right. Now, when it does this, okay, and I'm going to run out of boxes here. So maybe I should have done it this way. I think maybe, let me go ahead and do this. I'm going to go ahead and move that over here. Okay. And I'll decrease it in size. Okay. And so I'm going to put an extra box here. And you say, oh, well, that's nice. Thanks a lot. You didn't allow us to make an extra box. That's right. I didn't. I didn't tell you to make an extra box. Okay. That's the prerogative that I have. Okay. But what it's going to do, and I'm just doing this to just keep it consistent with the flow chart over here, phosphoprotein phosphatase is going to do this. It's going to remove the phosphate on PFK2 FBPase2. Okay, so what we're going to have here is this PFK2. Oh, I don't want to use blue because I use blue over on the other side. I'm going to keep the red. Okay. Oops. There we go. So PFK2. Oops. Boy, I'm making all sorts of mistakes. PFK2, there we go. FBPase 2 will be dephosphorylated by the phosphatase, the phosphoprotein phosphatase. That's what phosphatases do. Phosphatases dephosphorylate things, okay? And so I'll just leave off the phosphate, and what we're going to do here then is say that, okay, this then, when it's dephosphorylated, becomes active, okay? Now that is going to then start to make fructose 2,6-bisphosphate. Okay, so what's going to happen here then is that we can say fructose 2,6-bisphosphate, instead of going down, is going to go up. Okay, so if you were to think about it this way, what this enzyme does, and the kinase domain of this does, is that it takes fructose 6-phosphate and makes fructose 2,6-bisphosphate. Okay, so that's going to cause this to increase. Notice that we have here an up arrow, right? Right. Now, just as I was saying over on the left side, right, when we look at, whoa, sorry about that. When we look at fructose 2,6-bisphosphate, it is an activator of PFK1 when it's high, okay, and it's an inhibitor of FBPase1. So we could say this, okay, we can write this down and say that PFK1 will be activated. Okay, now if you remember what PFK1 does, well, PFK1 does this. It's part of glycolysis. It's going to take fructose 6-phosphate and make F16BP, fructose 1,6-bisphosphate. It takes ATP to do that, if you remember. Okay, and this is the third step in glycolysis, and of course that's going to go on to the rest of glycolysis. So we can say here in our last little presentation here is that glycolytic flux increases. Okay, and that's all in response to insulin signaling. Now, you might think to yourself, well, why does glycolytic flux need to increase in response to insulin, which is in response to a rise in blood glucose? Well, the reason being that then the liver will start to absorb more glucose, and it will start to then take that glucose and make other things from it. In particular, we will find out as we get into lipid metabolism, insulin at the same time is going to be activating lipid biogenesis. We're not there yet. We haven't talked about lipid biogenesis, but we'll get there at some point in time. And so then when blood sugar goes up, blood glucose levels go up, insulin activates glycolysis so that then the glucose levels in the liver cell will start to go down, so that then the liver will pull in more glucose, and then some of that glucose will be converted all the way through glycolysis to pyruvate, and then from pyruvate it will be converted to acetyl-CoA, and acetyl-CoA will then be used to make lipids. Okay, the other aspect of this will be that some of that glucose will be used to make glycogen. Okay, and as a store of glucose. Okay, and of course we talked about glycogen synthesis pathways as well. Okay, glucagon on the other hand will cause glycogen to be broken down. Okay, and that will be something that we'll look at later. Okay, all right, so that's then activity number 1, okay, and hopefully this gives you some like, oh, okay, so this is how I organize these things in my mind. I would encourage you to do this for other, regulatory mechanisms in other tissues. I would encourage you to do it for glycogen breakdown and glycogen synthesis, okay, so just think about doing those things, okay. I think this will be very helpful. Okay, now activity 2 was create a comic with the following parameters, okay. You can most certainly do that. I'm not going to do it right now. I'll go ahead and release a couple of cartoons that have been done in the past, okay, so maybe that will help you out. You can look at it and laugh at those kind of things, and also it will give you some a different way to study this stuff, okay. And that really honestly is what you have to do in a class like this. You really have to kind of make these logical structures in your mind of these concepts, and quite honestly making comics and doing those kinds of things really cement that information in your mind, so long as of course you make those right. Okay, all right, so hopefully you found this helpful. By any means, if you have questions, let me know.